Hey there, my name is Omar Ibrahim, and today I am going to be talking about PCR. The objective for today is to talk about what PCR is and how is it implemented today. So what is PCR? PCR, also known as polymerase chain reaction, is a common laboratory experiment technique used to make many copies of a particular region of DNA. Typically, the goal of PCR is to make enough of the DNA region that can be analyzed or used some other way. For instance, DNA can be amplified by PCR and sent for sequencing, visualized by gel electrophoresis, or cloned into a plasmid for further experiments. For PCR to take place, certain ingredients are needed: Taq polymerase, primers, template DNA, and nucleotides. We will be going over all of these ingredients and how they affect PCR and why they are needed. Since we're going to be reaching high temperatures in PCR, an enzyme is needed that is heat tolerant. This enzyme is Taq polymerase. Taq polymerase is a DNA polymerase enzyme that makes new strands of DNA using existing strands as templates. Taq polymerase can only make DNA if it is given a primer. A primer is a short sequence of nucleotides that provides a starting point for DNA synthesis. And so a pair of these primers are binded to the template. They can be extended by the polymerase, and the region that lies between them will be copied. The basic steps of PCR starts with denaturation. The reaction is heated at 96 degrees Celsius to separate or denature the DNA strands. This provides single-stranded template for the next step, which is annealing. After the heating of 96 degrees Celsius during denaturation, it has to be cooled to 55 to 65 degrees Celsius. The reaction is cooled so the primers can bind to the complementary sequences on the single-stranded template of DNA. And then next is extension. We raise the temperature again to 72 degrees Celsius so the Taq polymerase extends the primers, synthesizing new strands of DNA. Now that we've talked about what the PCR is, how do we implement what it is today? Well, for the COVID-19 PCR test, genetic material is extracted and then targeted for the specific gene segments that it only has. And then we make millions of copies to detect if the virus is present or not. If it is positive, that means you do have the virus, and if it is negative, it means that you do not have the virus. PCR can also be used in crime scenes. DNA extraction is copied and then cut by restriction enzymes and then placed into wells, and also a special gel named agarose. By process of gel electrophoresis, the DNA fragments are separated by size, and then they are placed into a nylon sheet. And the lines produced by the DNA can be compared with those of the other suspects.